Hello, everyone. Welcome. Thank you for joining us. Oh, I'm just making sure the video is going through. If we could do that quick spotlight. There we are. Thank you. Hi, everyone. So wonderful to see you. Welcome to our webinar, Probing Functional Autoantibodies in Patients with ME/CFS. I'm Emily Taylor, the President and CEO here at Solve. Thank you so much for joining us today for this free educational webinar where we will discuss some exciting upcoming research. Just this past May, in honor of World ME Day, here at Solve, we launched a new research program that was an important part of my vision when I stepped into the role of CEO last year. My goal with our Solve ME/CFS Catalyst Awards was to support promising research projects that have already made significant strides in advancing our understanding of ME/CFS, long COVID, and associated conditions. While our Ramsey Research Grant Program targets early career scientists and novel research approaches, I hope to deliver a Catalyst Award program that accelerates existing project results. Like the catalyst of the chemical reaction that is their namesake, these awards bypass barriers and accelerate progress, providing targeted funding to help outstanding studies reach completion, publication, or the next stage of investigation faster. To learn more about the Catalyst Awards or to donate to all of our research programs, please visit our website, and you can see that at solvecfs.org. So it's incredibly fitting that our inaugural ME/CFS Catalyst Award winner was Dr. Akiko Iwasaki, a pioneer in her field and our guest today. Her study, Probing Functional Autoantibodies in Patients with ME/CFS, is a vital step to uncover the biological drivers of ME/CFS and long COVID. This study focuses on a crucial and emerging area of investigation, the role of autoantibodies, antibodies that mistakenly target a person's own tissues. In the long-term neurological symptoms seen in patients, these have been related. In Dr. Iwasaki's lab will investigate whether these autoantibodies can actually cause symptoms such as fatigue, pain, cognitive issues, or motor dysfunction by introducing patient-derived antibodies into mice and analyzing their effects. The team will also use an advanced technology called HuProt to scan all human proteins to find which ones are targeted by specific autoantibodies. Autoantibodies against specific human proteins may be effective biomarkers, measurable features that can help us diagnose patients quicker or customize how we treat each patient. This kind of translational research is essential to turning biological discoveries into real-world therapies, and we are so proud to drive that work by funding Dr. Iwasaki as an inaugural Catalyst Award winner. For those of you who may be unfamiliar with her work, Dr. Akiko Iwasaki is a Sterling Professor of Immunobiology, Professor of Dermatology, and of Molecular, Cellular, and Developmental Biology, and of Epidemiology of Microbial Diseases. She serves as the Director of the Center for Infection and Immunity at the Yale School of Medicine. Dr. Iwasaki is an investigator of the Howard Hughes Medical Institute. She was president of the American Association of Immunologists from 2023 to 2024, and has served as an AAI council member from 2018 to 2025. Thank you so much for joining us, Dr. Iwasaki. I'm so excited to have you join us today and share more about this really exciting work in autoantibodies. And again, cannot emphasize enough how you are pioneering this field, and we are so thrilled to work with you. So would you be so kind to share a little bit about your Catalyst Award-winning study?
Thank you so much, Emily. Can you hear me?
Loud and clear.
Okay, wonderful. I'm so delighted to be the inaugural winner of the Catalyst Award by Solve ME. It's such an honor for us to be receiving this support to be able to continue our work on post-acute infection syndrome in general, but particularly this award funds our probing functional autoantibodies in patients with ME/CFS. So today, what I'd like to do is to give you some update and research insights that we're gaining from long COVID, and to be able to apply the same kind of approaches to studying ME/CFS. So can I share? Yes, I'm going to share my slide. Okay, can you see my slides?
Perfect.
Okay, wonderful. So this study is an ongoing study that's that we are probing the role of these autoantibodies in long COVID patients, but the insights we gain from this study will be directly applicable to other post-acute infection syndromes and as well as ME/CFS. So before I begin my talk, I just wanted to remind everyone that there are many other medically unexplained syndromes that are very similar to long COVID and ME/CFS that can happen after infection with a variety of pathogens, including Ebola, dengue, polio, SARS, chikungunya, EBV, and many others. And some of these conditions don't even have its own name. But we know that their previous studies have highlighted the possibility of developing similar kinds of chronic conditions after these types of infections. And there are also non-viral pathogens, bacterial and parasitic pathogens that have been reported to cause similar chronic conditions. And this review was a product of a collaboration with Jan Chotka, who himself have ME/CFS, and he's a patient researcher, and he's been studying these conditions for many years. And we were very fortunate to collaborate with him to learn more about previous evidence of these types of conditions. And Mehdi Hornig and Vivraj Jansari was a summer student who also helped with this article. So the reality of these post-acute infection syndromes are quite dire, that just long COVID alone, the cumulative incidence globally is estimated to be 400 million people. And even prior to that, 65 million people are estimated to be suffering from ME/CFS pre-pandemic. So if you combine these numbers, plus there are daily sort of increases in the people who are getting these types of conditions as a result of infection, this is really a dire problem throughout the world. And some of the statistics, for example, that over a quarter of ME/CFS patients are bedbound or housebound, and these conditions can strike people of all ages and backgrounds. It is really a reminder that we need to prevent these diseases from happening in the future. And not only is it a social and economic problem, but also a national security problem, as military personnel are also affected by these conditions. So these post-acute infection syndrome are can affect anyone, but in fact diagnosed more often in women. And people of middle age are at higher risk of developing these conditions. However, it's very important to remind ourselves that even young children can get these post-acute infection syndromes, whose symptoms may not be readily recognizable unless the physicians are aware of what to look for. So there's a lot of ongoing emerging research on pediatric long COVID as well. So I'm a basic scientist and immunologist, and my lab studies sort of fundamental causes of infectious diseases. And we think that there are at least 4 possible root causes of post-acute infection syndrome, such as pathogen reservoir that can be established after the acute phase. So this is the inability to completely clear the pathogen that could be contributing to disease. There's also autoimmunity that can happen after an infection. This is a well-documented phenomenon that infections are linked to the development of autoimmune diseases, because infections can trigger autoantibodies and self-reactive T cell stimulation as a result of molecular mimicry, which means that the pathogen antigen mimics the host protein, and therefore the cross-reactivity can happen. It's also possible that unrelated antigens can also become target of autoreactivity as a result of being in the vicinity of the inflammatory sites. There's also tissue damage and dysfunction that can happen. We know very well that during the acute phase of COVID, there's a lot of vascular issues as well as coagulation that can damage the tissue, which is very difficult to restore. And inflammatory signals alone change and reprogram the cell types such as microglia that can have a lasting impact in the brain. And then there's also latent virus reactivation, which can occur in definitely a subset of people with ME/CFS as well as long COVID, where Epstein-Barr virus and other herpes viruses can reactivate in these people after the acute infection. And that could also contribute to autoantibody responses, tissue damage, and inflammation. And these things are drawn as cogs of a machine sort of driving each other, so that it's possible that a person starts off with a viral reservoir that then triggers the autoimmune disease, which causes tissue damage, reactivating herpes viruses, or vice versa. It could start with the herpes virus and so on. And in some people, all the 4 gears are turning. In other people, maybe only one of these things are activated. So the critical goal of the field is to be able to identify patients that have these distinct drivers of disease, and so we can properly treat these patients with medicines and drugs that are available in that field. So I'm going to give you some sort of experimental evidence to support that there are these 4 root causes are happening in people, and focus on the autoantibodies, because that is the research that's supported by SolveME for the ME/CFS patients. So this is a study that we initiated with Dr. David Petrino at the beginning of the pandemic in 2020, where we decided to put our forces together to collect blood samples and saliva from patients with long COVID, and to be able to analyze the biological changes that occur in patients with long COVID. And so Jamie, Laura, and Dana from David's team contributed significantly to the study, as well as the Yale trainees who led the study, John, Rahul, Peiwen, Jill, Jeff, and Sasha. And Aaron Ring and David Van Dyke provided their expertise in serology analyses as well as computational approaches. So with this Mount Sinai Yale Long COVID study, or My Long COVID study, we began to analyze biospecimen from patients who have long COVID, as well as convalescent control and healthy control participants. And we also had external long COVID cohort to validate the key findings. And from these patients, we collected blood and electronic medical record and symptom survey to be able to identify key biological features that are distinct between long COVID patients and convalescent controls and healthy controls. Here we looked at immune cell types using flow cytometry, looked at antibody responses to SARS-CoV-2 antigens, as well as against virtually any antigen that we wanted using peptide display library, as well as some autoantigens, and did some plasma proteomics. So the age group of this cohort was between roughly around 30 to 50 years of age. The sex is female dominant. As I mentioned, female sex is a risk factor for developing these post-acute infectious syndromes. And we also focused on patients that were not hospitalized in order to reflect that 90% of the patients with long COVID start off with mild COVID infection in the acute phase. And days from acute COVID was around a year from infection. And these cohorts kind of provided some early insights as to what cells are being impacted in patients with long COVID. For instance, when we looked at the T cell subsets, we see that PD-1 positive, TIM3 positive, exhausted T cell phenotype percentages were elevated for CD4 and CD8 T cells, as well as reduction in the central memory T cell subsets, the CD4 subsets here, suggesting that these T cells are continuing to see the antigens without being able to clear the antigen. And whether that's an autoantigen or viral antigens, we don't know what the target is. But Nadia Rohn's team subsequently published that her SARS-CoV-2 specific CD8 T cells also have the exhausted phenotype in patients with long COVID, suggestive of potentially chronic antigen presence in these patients from the virus. And when we stimulated these T cells in vitro using artificial stimulant, we were able to detect that the ability to secrete cytokines, which are important for communicating between T cells and other cell types, are elevated in patients with long COVID, including type 2 immune cytokines such as IL-4 and IL-6. And this feature was pretty much only detected in patients with long COVID. Looking at their antibody response to SARS-CoV-2, we see elevation in antibody response against the spike protein, the total spike, the S1 region, and the receptor binding domain, suggesting again that there may be a chronic presence of antigen that's driving the elevation of these antibodies. And for this study, we only included patients that had 2 doses of vaccines to normalize the exposure to the antigen. And yet we see significant changes between the convalescent control and long COVID. Again, suggestive, it's not a direct evidence, but suggestive of a presence of antigen, a chronic antigen from the SARS-CoV-2. And when we looked at the plasma factors, we saw elevation in immune factors, inflammatory factors such as complement and chemokines and cytokines from patients with long COVID. But what really stood out in this analysis is the reduction in the morning cortisol level. And this was so striking that we wanted to validate this in a separate cohort. So we collaborated with the Yale Pulmonary Long COVID Clinic to collect patient samples from a separate clinic. That's what this external long COVID cohort is. And those patients also exhibited lower levels of morning cortisol compared to the healthy control and convalescent control. And cortisol is a very important hormone that controls many physiological functions, such as wakefulness, motivation, and many other phenotypes. And this it's a diurnal hormone that elevates during the morning and it declines during the day. And so it was very important to ensure that the collection time did not differ between the different groups, which it didn't. And cortisol, as many of you know, is an important hormone secreted by the adrenal glands in response to its central nervous system signal, the hypothalamic corticotrophin releasing hormone acting on the pituitary glands, which secretes the ACTH hormone, which then acts on the adrenal glands to produce the cortisol. And once it's produced, it has a negative feedback loop to stop the further production. So the level of cortisol in circulation really controls its own level through the regulation of central command coming from the hypothalamus and pituitary. So because we see this reduction in the level of cortisol from patients with long COVID, we wanted to understand if there is a compensatory increase in ACTH level in order to try to elevate the low levels of cortisol in these patients. However, when we measured the ACTH level, there was no increase coming from the ACTH in the long COVID patients, suggesting that the deficit may be upstream of the adrenal glands in the hypothalamus or the pituitary. And this also became obvious when we were studying sex disaggregated data from these patients, showing that testosterone and estradiol levels were also impaired in a subset of long COVID patients. And in terms of other hypotheses that I mentioned, such as the latent virus reactivation, we examined using Dr. Ring's rapid extracellular antigen profiling strategy. And here what we noted, so these graphs are basically representing the score of antibody reactivity against the antigens that are indicated on the x-axis here from patients with long COVID on the right and healthy control and convalescent control on the left. So the darker diagram indicates the distribution of patient antibody levels against a particular antigen. And here what we find is that antibody against Epstein-Barr virus GP42 and P23 are elevated in patients with long COVID. So is the antibody against the VZV. This is a chickenpox or shingles virus, glycoprotein E reactive antibody are also elevated in patients with long COVID. And this is despite the fact that the seropositivity, meaning that latency established in the long COVID versus control, were similar between these volunteers. So these elevation in the lytic antigen reactive antibodies are suggestive of a recent reactivation with EBV or VZV. And this is something that we are continuing to investigate, whether these patients, there may be a pathological driver contribution from these reactivated viruses to long COVID. So if you look at, David Van Dyke's team did a machine learning analysis to determine a handful of immunological factors that can distinguish patients with long COVID with a 94% accuracy. And here, what they found were these factors such as the lower levels of cortisol, lower levels of dendritic cell type 1. These are the cells that educate the T cells to become cytolytic, as well as reduction in the central memory CD4s and CD8s that I mentioned earlier, and the increases in things like galectin, EBV reactive antibodies, and CD4s, and some chemokines. So this really showed us that these handful of biological factors alone are able to distinguish patients with long COVID, and that these may be a very important clue as to what might be driving the disease in patients with long COVID. So, so the main topic of today's seminar really is to focus on the autoimmunity. And this was a project that's really being led by talented postdoc Kayla Sa from the lab, and in collaboration with other trainees, Julio and Rafael, as well as with Dr. Petrino and Tomas Horvath, to really understand if there's a causal link between autoantibodies and neurological symptoms in patients with long COVID. And the approach that we took was to first try to analyze whether patients' antibodies, the purified IgG from healthy control, convalescent control, or long COVID patients that I've already described from the Mount Sinai Yale long COVID collaboration, whether those antibodies bind to tissue sections from patients. These are just tissue sections that were obtained from patients that are not related to long COVID, but from the biobank that we have at Yale. And what we noted was that there were some specific staining pattern that we observed with long COVID patients' IgG. I hope you can see my cursor here, where you see this green staining in the tissue that indicates human IgG bound on the section. And this is from a section of human locus coeruleus. And you can see also that the mean fluorescence is elevated from patients, especially in this case, female patients with long COVID. And these selective staining is seen within this tissue section, as well in the human thalamus, where you see this very clear staining pattern from IgG obtained from patients, but not the convalescent controls or healthy controls. And you can see this numerically listed here, that there are certain subset of patients that have reactivity with their antibody against specific targets within the human brain. And we also tested mouse tissue sections and found that, again, certain tissues such as the sciatic nerve or the meninges have a positive stain with IgG isolated from long COVID patients. You can see very clearly the green staining here and here. And again, it's not that everybody has staining, but there's a subset of patients here, particularly in this case, female, but also male patients that have reactivity to structures within these nervous tissues. And in some cases, we can even identify the target cell types. Here, what we are looking at is, again, IgG isolated from long COVID patients staining the mouse meninges. And here we are also co-labeling structures like the endothelial cells with the yellow signal CD31, as well as the pericytes stained in red here. And if you take a cross section of this meninges stained with the human IgG, you can see that there's a significant overlap between particularly the pericytes, but also other structures around the vessel. So this suggests that at least from this particular patient, there is this staining that you can observe that is specific around these vasculature. And interestingly, when we look at the mouse meninges stain, we can actually correlate those antibodies that stain positively coming from patients reporting headache here, for example. So there may be some link between the reactivity to mouse meninges and some of the symptoms that are reported, such as headache. So the critical question is, what are the molecular targets of these autoantibodies? And to do a unbiased screening analysis, we used the CDI laboratory HuProt assay that Emily mentioned earlier, where we purify the IgG and then we stain a microarray of slides that are sort of spotted with different human proteins. And there are 21,000 full-length, correctly folded human proteins that are printed on these arrays. And we add the IgG, and then we add the secondary antibody to see where the IgG bound. And we can see that there are some spots, for example, in this diagram, that light up with specific patients' antibodies. And so we did this for the healthy control, convalescent control, and long COVID patients. And what we found is that the patients with long COVID had, in general, higher number of autoreactive antibodies per patient compared to the convalescent control or the healthy controls. Now, everyone has some reactivity to different autoantigens, and they're not all pathological. But the fact that we're seeing a significantly higher levels of autoantibody counts, it's very interesting. This may indicate that some of these autoantibody may have a pathological function. And this was true in the case of wherever we basically cut the Z-score threshold. So essentially, this is a very robust increase in the number of autoreactive antibodies from patients with long COVID. And to get a little bit more granular insight into this, for instance, if we selected dozens of autoantigens that are known to be CNS tissue enriched, we can see that the long COVID patients, again, like everyone has some kind of autoreactivity, but the higher yellow here indicates a higher reactivity to specific antigens. And you see that long COVID patients have this smattering of these autoantibodies compared to the healthy control and convalescent control. And there are some basically autoantigens that have been found in other autoimmune disease conditions, which are listed here, that also are found in patients with long COVID. For example, this particular patient, LC66, has reactivity to the Smith antigen, which is a well-known autoimmune or autoantigen found in systemic lupus erythematosus. And there are also other types of autoantigens well characterized in other autoimmune diseases that are being found from patients with long COVID. Now, there are some reactivity from the control groups as well. So we can't say that this is really a pathological level of autoantibody, but at least we're finding some of these things. And if you extend this sort of reactivity to larger set of autoantigens, you can start to see that lots of patients with long COVID have, again, large number of autoantibodies that are reactive against many, many antigens. So there isn't like a single public autoantigen that everyone with long COVID is reacting to. Not at all. It's a collection of autoantibodies that are elevated in patients with long COVID. And these are, for example, more frequent reactivity to the CNS-enriched autoantigens. So we also can correlate the number of symptom burden that's reported by the patients or the number of organ systems that are involved against the log count. This is the number of autoantibodies or the magnitude of the autoantibodies that are found in patients. There seems to be a positive correlation with the autoantibody number and the magnitude of symptoms and organ systems involved. And so with this insight, we decided to do a passive transfer of antibody into just regular wild type mice. So each mouse is injected with purified IgG from patients or controls. And we then follow the behavior of these mice. There are multiple behavioral tests that we did: open field, elevated serum maze, rotarod, grip strength testing, hot plate test, blood pressure, heart rate, and collected blood and tissue for further analysis. And just to confirm that the purified IgG is a pure monomer of IgG and there are no other sort of contaminants in the isolate, we ran a size exclusion chromatography and it looks very good. And we can also measure the amount of human IgG in the recipient mice over time, and they're pretty stable over time in the serum, and there's a little bit of that antibody access to the brain, which may explain some of the phenotype that we see. So after following all of these behavioral studies, we came to this one assay which showed significant changes in IgG isolated from patients with long COVID, which was the hot plate test. So this is really looking at the number of seconds it takes for the mice to react to a heated plate, 55 degrees Celsius. And this is a standard protocol to measure increased thermal pain sensitivity. And what we see is that in mice that are injected with saline, this is the sort of the baseline latency to behavior is about 10 seconds. And healthy control IgG is about the same. And convalescent control IgG is also about the same. But when we look at the mice that have been injected with long COVID patients' IgG, we start to see a reduction in the latency to pain behavior, meaning that these mice react much quicker to the thermal sensation. And when we break down the patient characteristics in this experiment to those patients who reported inflammatory or neuropathic pain compared to the no new onset pain, you can see clearly that patients with chronic pain conditions are the ones who have IgG that can possibly transfer the pain phenotype in the animals. So how does this happen? Chris Baker, who's a neuroscientist in the lab, used a reporter mouse system to be able to identify the changes that occur after injection of IgG from patients. And so we can do this longitudinally using a reporter mice that have a nociceptor neurons that are labeled with TD tomato. This is a fluorescently labeled neurons that you can measure in the skin. And what Chris is doing is to monitor the number and the volume occupied by the intraepidermal nerve fibers in the skin of the animal that are injected with IgG from patients or control. So doing this analysis, it was very obvious that mice that are injected with long COVID patients' IgG started to lose the intraepidermal nerve fiber volume as well as the number of fibers in the skin. As early as 1 day post-injection, we can start to see a decline in the number of fiber as well as the volume of these fiber. This is sort of depicted here that, again, these fiber numbers or volume here is particularly impacted in the mice that received IgG from patients reporting chronic pain. And we also see that when you take the blood from these mice after the experiment is concluded, we see that there's an elevation in the neurofilament light, which is a biomarker that's used to detect neuronal damage, because this is a neuron-specific protein that's released only upon damage of the neurons. And you do definitely see this increase in the plasma level of these NFL protein in animals that were injected with IgG from long COVID patients. So what's happening to the entire brain? Well, this is a great collaboration we have with Mark Schneeberger Payne's lab at Yale, where he does these whole mount activity maps following passive IgG transfer into mice. And the way we do this is we collect the brain from the mice that are passively injected with antibody from patients or controls. And then we do this clearing method to be able to visualize neurons that are activated through the CFOS labeling. And here, what we're seeing is the somatosensory cortex, anterior cingulates, and dorsomedial hypothalamus. These areas that are lighting up, that really includes this pain circuits in the brain, as well as potentially hypothalamic control. And we also see this in other areas of the brain as well. And so we're now trying to understand how IgG reactivity that we know from the HuProt assay, how they are kind of correlating with the activation of different neuron types within the brain. So to summarize our findings, we know that patients with long COVID carry antibodies that react to multiple human neural tissue regions, and that these antibodies have many specificities. It's not a unique one public autoantigen, but multiple specificity, including some common autoantigens, as I mentioned before. But every single patient seemed to have their unique pattern of specificities, some of which include the CNS antigens. And that passive transfer of IgG is sufficient to induce the pain behavior, muscle weakness, and loss of balance. This, I didn't have a chance to talk about these, but some subset of patients can also confer these phenotype in mice. So what do we do for the for our next step for this long COVID? We want to continue to actually identify autoantigens that are now sufficient to recapitulate pain and peripheral neuropathy. We know that the collection of antibodies can do this, but what are the exact autoantibody that confer this pain sensation? And we will do similar passive IgG transfer study from other post-acute infection syndrome and ME/CFS. And this is in line with what I told you about the SOLVE-ME supported research. And we will do, we would like to, based on those understanding, conduct a randomized clinical trial to test various autoantibody targeted therapies, such as FcRn inhibitors, which basically reduce the level of autoantibodies in circulation. There are many B-cell targeting therapies that are used in autoimmune diseases that we might be able to try out in patients with ME/CFS or long COVID, or transferring IVIG, which is many patients are already receiving IVIG for a variety of conditions, but a randomized clinical trial might reveal the effect size of these benefits. In any case, there are many sort of great medicines available in the space of autoimmune disease, which may be able to, we can sort of retool it to identify if they're beneficial in a subset of patients with these autoantibodies. So specifically, the SOLVE-ME supported research will allow us to start to identify these autoantibody targets in patients with ME/CFS. We can analyze the reactivity of antibodies against tissue sections, as I showed you for the long COVID, and to conduct passive transfer of these antibodies and to see whether that confers behavioral phenotype in mice. And that will also inform us which organs are maybe targeted by the antibodies. And so I have a great postdoc, Kenji Ota, who is starting to analyze these issues in-house. So I want to end here by thanking the incredible people who I get to work with every day, who are advancing these insights, both from my own laboratory, as well as I mentioned already, Dr. Petrino's team. And then we also have a separate cohort of recovery and lesson study with Dr. Harlan Krumholtz. And we just had a publication with Swedish ME/CFS cohort, cerebral spinal fluid analysis with the Brage Clinic in Sweden, as well as with Michelle Monge. We're continuing to analyze how inflammation in the CNS contributes to cognitive decline. And we obviously thank SOLVE-ME and other foundations for the support. So thank you so much for your attention.
Thank you, Dr. Iwasaki. That was fantastic. I have had the privilege of reading your work before, but hearing you present it and seeing it in context, it just strikes me how much clicks with what we already know about ME/CFS, the reduced cortisol levels, the cytokine, like all these things, and they just fit. And so it's just so exciting. I'm so thrilled to hear about this work. The questions have been pouring in. Folks are very excited to hear more, but I'll start with some of the questions that were submitted in advance of the webinar from your registration forms for folks who joined us today. The first question I'd like to ask is, wow, are there any current treatment pathways that your work suggests could be effective for patients with long COVID or a similar ME/CFS? For example, would IVIG be effective to outcompete antibodies? Or some folks in the chat also mentioned rapamycin for immunomodulation, or SCIG was another one that was mentioned. So are there any current treatment pathways that are being explored that would coincide or overlap with your work?
Thank you so much for that question. Yes, I kind of mentioned this a little bit at the end of the talk, but there are very promising medicines that are available to reduce the level of autoantibody. So I'm not suggesting that those approaches will work for everyone. Again, we need to be able to identify people who have autoimmunity and target that specific subset. But if we can do that, yes, absolutely, we should be doing randomized clinical trials for IVIG and rapamycin. We're actually doing one with Dr. David Petrino's team right now. Yeah. Yeah, this is with the long COVID patients, but obviously any insights we gain in one population may be informative in another. So we are currently actively recruiting patients for rapamycin trial, as well as I mentioned, B-cell depleting therapies. There are gradients of monoclonal antibody that deplete B-cell, different B-cell subsets, lightly or deeply or completely. And those things are, having some incredible impact in autoimmune diseases. So I would love to see or be involved in a trial where those monoclonals are tested as well. Yeah.
Yeah, I'm noting that for future funding decisions. That was actually one of the questions you just answered was what are some insights or some follow-up that could be done to generate further insights? And that's you've already answered that question. That's exactly where I think we'd like to see this work go next. With all of this underway, if obviously a big if we find the same results for ME/CFS patients that we found for long COVID patients, would you say that means these diseases are autoimmune in nature? Does that answer the four-piece conundrum, or is it still a combination, or what would that suggest?
Yeah. So as you saw from my data, it's not that all the patients have elevated autoantibody, right? So, but if we look at the subset of patients who have number of autoantibodies and who can transfer the pain phenotype, for example, that's like to me, that's a causal relationship. You take something from the patients and you confer the same phenotype in mice, right? So that is a direct sort of link between the autoantibody and disease, and that's sort of the phenotype or the subset of patients that I think are benefit the most from these kinds of autoimmune treatment. So, but the critical thing, as I mentioned earlier, is the coming up with some sort of a panel to be able to detect who has it, because we can't transfer everybody's antibody in the device, right? And study that. So, HuProt is one of the potential mechanism of identifying patients with very high number of autoantibodies or CNS enriched antibodies. That's something that we're exploring.
Absolutely. And so that could be, dare I say, that mysterious biomarker that we're seeking.
Yeah.
And is that test widely adopted? Kind of what is the status of that level of test in terms of a bench to bedside translation? Is it still a research only space, or what would you say would be the magic timeline if we could get more funding, right? To make that possible.
Thank you, Emily. Yeah, definitely. I don't want to promise any timelines or anything, but
Of course, of course.
yeah, but you, as you say, it's one of the promising path towards identifying that subset of patients, right? So HuProt has been collaborating with us to be we want to be able to share these data like across public whoever interested to be able to compare for example long COVID ME/CFS against lupus or arthritis or some other conditions, so that we can start to gain idea about what are the patterns of autoantibody reactivity across different diseases, and can we use some sort of pattern to come up with a biomarker for say post-acute infection syndrome that's driven by autoantibody. So we're trying to chip away at that, and hopefully, like if others agree, also to share these once we're published this, publicly available, then we can start to compare across many different diseases, because CDI has analyzed obviously many different conditions like cancer, Alzheimer's, and other things. So I'm hopeful that if we can put our efforts together, we may be able to identify patterns.
Wow. Wow. That is so exciting. And so thinking, I always love to think that next step bigger. So would this data potentially be able to illuminate common comorbidities with these conditions? Like, for example, we know that about 33% of folks with endometriosis often overlap with ME/CFS. So again, assuming this replicates in ME/CFS and assuming all the data comes together, is that a potentiality of this work?
Yeah, I would love to be able to do that, Emily. Like to be able to identify markers of comorbidities that lead to that are more enriched in these conditions, for instance. And autoantibody may be that, or it could be like co-infection with other pathogens. So we're not like just restricting our brains to just autoantibodies, but other drivers that may be driving two diseases in a patient. Of course, there's the genetics, right? Like that I didn't even touch on, but there is definitely genetic predisposition to developing either the comorbid conditions or the, ME/CFS plus the endometriosis, for example. So I think the next level would be to look at the genome of these patients and to be identify a molecular level, like which genes may be contributing. Yeah.
Wow. I'm going to turn to a couple more questions that came in through the chat. We have so many that came in. There's been a couple. Oh, this is an interesting. I'm going to try and combine a couple questions into one. So one questioner points out that this is very interesting and very relevant for folks who have a clear infectious trigger. But what about our community with the folks who have like a gradual onset or can't trace their infectious trigger? And then continually, does this potentially account for other types of onset? So those are kind of two questions I smushed into one.
Yeah, no, that's a great question. So, for the non-infectious triggered ME/CFS, those 4 wheels that I showed you, the first wheel is gone. We don't know what it was the trigger, but it's not the infection, right? But we still have the autoimmunity wheel, we have the inflammatory wheel, and we have the latent herpes reactivation wheel. And those could be turning in those patients. Now, we don't know what exactly caused those wheels to turn, but autoimmunity is one condition that I can imagine could be shared in ME patients that have a non-infectious trigger. So, autoreactive B cells and T cells can become activated in the absence of infection. It could be trauma, it could be other conditions that promote that, or genetics, of course. So I think,
Like environmental triggers, for example.
Yeah, exactly. Environmental triggers, yes. So that's the one wheel that I can see, common to many different triggers. But again, we need to do the research. Yeah.
Wonderful. Well, I know gosh, I'm just trying to catch up on the questions. I think we answered this question on rapamycin, but there was the question specifically talked about subgroups for rapamycin. So would this work or some of the other work that you're doing in the rapamycin space suggest that there could be a test to treat model for some of these subtypes?
Absolutely. So there was a really nice paper by Professor Mark Davis from Stanford recently, where in the proceedings of National Academy of Sciences, where his group showed that lymphocytes from patients with ME or long COVID, they had elevated reactive oxygen species that you can measure using flow cytometry. And those are the conditions that I think would benefit from rapamycin, because rapamycin can induce autophagy or mitophagy to restore damaged mitochondria and hopefully remove the reactive oxygen species coming from the mitochondria. So that's one sort of subset that I can see benefiting directly from rapamycin. But rapamycin, as you know, has many different impacts, some of which are on the immune system, others on different organ systems. So it's hard to kind of predict who might benefit. But, since patients with ME/CFS treated with the rapamycin, some of them do really well on the low-dose rapamycin treatment. And so we want to capture the biological changes that happen with this treatment before the treatment, during, and after. And that's what that trial that Dr. David Petrino and I are engaged in right now. And so we want to gain biological insights from that trial to be able to say exactly what you're asking, Emily, like which patients would benefit from that.
Love it. Love it. That actually touched on another question that somebody asked in the chat, which was regarding the long-term impact of this understanding. Does this mean we're no longer neuroimmune and we're autoimmune? Like, well, how do we categorize ourselves now? Are we at a point where we change categories? Are we not there yet?
No, in my mind, it's all immune.
It's all immune. I love that.
Yeah, I mean, it's really it's about like the immune system, whether or not it's insufficient immune responses that can clear the pathogen, so that would trigger the persistent virus infection, or overt immune responses against self-antigen, which would trigger the autoimmune bit. And even when we say neural, that can be, we've shown and others have shown that that can be triggered by the inflammation, which is immune. So essentially, you can't really think about these diseases in the absence of the immune system. And they're contributing either positively or negatively in both ends, right? So I think that's why we believe that identifying the immune factors in the disease is so important. And I also wanted to kind of highlight, that we don't exist in a bubble. I mean, we obviously learned so much from the pre-existing literature from ME/CFS, and people like Carmen Scheibenbogen, who've been studying the autoantibodies in ME patients for many, many years. We learned so much from her and her team. And similar kinds of pain sensation transfer has been reported by the Amsterdam group, Jeroen van Dunen's group. So I want to acknowledge that, we're not the only one to do this research, but we're kind of building on the contributions of others.
That's so fantastic. And Dr. Carmen Schadenbogen is a Ramsey Award recipient. So we're so pleased to see all of those pieces coming together. I know we just have one. So I'm not enough time left for all the questions, but I'm just going to ask this last question, especially since we were talking about the immune nature of these illnesses. So we see differences in immune systems between men and women. Is that driving the patient population data that we see that predominates women? Is that something that would help us understand on the treatment side, maybe testosterone or some other kind of hormonal treatment to balance that out? What do we think? What do we take away from that big picture question in 1 minute or less? I'm sorry.
Oh my God, Emily, you need to invite me again because I have a whole story on that. But
oh wow, okay, well, next webinar, that topic. Okay.
No, but it's so true. It's very important to analyze sex disaggregated data because there we are getting insights about dysregulation of sex hormones in testosterone in female and estradiol in male. And that is also informing potential treatment for males and females.
Wow. I want to ask you so many questions, but we're going to have to wrap up for now. I thank you everyone so much for joining us for this fantastic webinar. Dr. Iwasaki, thank you for your time. Congratulations again on your Catalyst Award. I do hope that if you're interested in seeing more about the Catalyst Award program, folks will consider joining us for targeting autophagy and a clinical trial of low-dose rapamycin and ME/CFS, long COVID, and IACCIs. That's our next webinar on September 4th, which will continue this coverage of these awards programs. Initiatives like the Catalyst Awards, like the Ramsey Research Awards, and even this free educational webinar are only possible with the support of people like you. Thank you so much for those of you who donate to Solve, and please help us grow these programs to continue the great work of Solve by visiting our website, solveme.org/donate, or you can use the QR code that'll be bouncing up shortly on your screen. Thank you again. In the next few days, you'll also receive an email link to the YouTube recording of today's session if you'd like to see it again. That'll also be available on all of our organizational social media accounts. And after this event closes, you'll receive a brief survey on your screen. If you take a moment to share your feedback with us, we love hearing your feedback. It's so helpful. For example, I changed my background because we received feedback that our previous Zoom backgrounds were too triggering for neurological symptoms. So thank you for that feedback. We can improve. So please let us know how we can always make these webinars better. We're always seeking to respond to your needs. With that, once again, Dr. Akiko Iwasaki, it's been a true pleasure. It is absolutely so thrilling to see your work, and I can't wait to see where you go from here. Hopefully, we'll find all the funding to get every corner of this disease mapped and understood. But until then, we will keep pushing forward. So thank you again, everyone, for joining us. May you have good rest and many spoons, and we'll see you at our next webinar in September. Take care, everyone.
Thank you. Thank you, everyone. Goodbye.